All right, so we are just about a full house here already. So we'll get started with the presentation. As you should be able to see on your screen, I should say a change of view using root imaging to expand your research by CID Bioscience. Online with me is actually our distribution manager, Susie Truitt. She'll be helping in the background, facilitating with the Q&A, as well as the overall structure architecture of this webinar. And as you're an attendee using the Zoom software, just want to give you a couple tidbits. If you go down, and it won't show up immediately, but when you scroll all the way down to the bottom of your screen, you'll see a bar pop up, and it should give you options like chat, questions and answer, as well as raise your hand if you'd like. So I'm going to give the entire presentation and a short video for this webinar, but at the end of it, we're going to leave plenty of time for questions and answers. Any questions, please put them in that Q&A box, and we'll be getting to them towards the end. Now, this webinar is being recorded, so if you have anything you'd like to see later on, if you want to come back to it, we'll be able to share this later, as well as if any of your colleagues need the information as well. So to get started, if I can, we'll be covering the CI-600 and CI-602. Now, the 600 has actually been around for about 10 years and is a in situ root imager. And the 602 has just been developed as of last year using more recent technologies, being able to diminish the size of the root imager, but still keep it so that it can be applicable out in the field in already established tubes. So we shall. But first, let's cover why should we study the roots. One, it's a critical success for the whole plant. They are absolutely critical for understanding plant or crop yield, environmental effects, the additions of certain chemicals and elements, and greater emphasis has been placed on root study that no longer are instruments such as the mini rhizotrons simply in the domain of scientists, but they're also being utilized by crop consultants, agricultural researchers, farmers, to understand the effectiveness of soil treatment, fertilizers, and other nutrient applications. They can be able to use for, say, measuring and planning for the impact of drought and temperature fluctuations, or monitoring for root pathogens and estimating crop yield. So root study is really, just an integral portion of the understanding of the plant's health and production as a whole. So getting back to kind of root study, a lot of studies are very much actually destructive of the roots, which really kind of has a hard effect to being able to visualize the roots in their natural biome, do a non-destructive assessment and see how they grow, and being able to do a repeated over time assessment of both growth, application studies, and pathogens. So since their proposal by the J.H. Bates back in 1937, mini rhizotrons have been constructed with a kind of a successive list of technologies, starting down with just mirrors, in order to visualize roots and provide means to non-destructively and repeatedly assess root conditions. So mini rhizotrons have been utilized to study, again, plant response to drought conditions, compare treatments over time, my personal favorite, map phenotypes to genotypes, and overall find correlations between below ground characteristics and above ground function. However, mini rhizotrons in the past have some major historical setbacks. In fact, even the rhizotrons that they came from, which were originally just very large boxes you would actually be able to walk into and have glass walls, have some issues you would have to deal with, including restricted accuracy, low image quality, limited size. I'd also like to put in here, very hard to replicate a very similar environment when you're allowing in other environmental factors like UV light. The great thing about modern systems, especially with the CI-600 and the CI-602, is that it's lightweight and portable. It's ideal for remote locations. You can use root tubes, glass to acrylic, and have variable DPI, so like the dots per inch, and that's just the resolution levels which you're scanning, so that you can do very quick scans at low resolution, getting your information, or you can do more intensive scans at higher resolution, so you can see things all the way down to root hairs and mycorrhizae. So the CI-600 To answer a lot of the problems that mini rhizotrons have had in the past, we've developed this over 10 years ago. It's portable, scanner-based. As you can see in the tube, the scanner is just attached to a sliding rod as well as a computer, which is off screen. And the tablet computer, with that, you can just press go and you can scan 100, 300, or 600 DPI. And using both the cap and the sliding rods, you're able to measure as you go down the tube visibly so you know how many scans you're at. And the great thing with using technology like this is that you can do one scan at one level, slide it on down at a measured rate, do another scan at a different depth, and then later on in our RootSnap software, you can actually take these scans and put them together to make an overall large picture of the root system that you're looking at in that location. We also actually provide, when we do give out the CI-600, we actually provide 6 acrylic tubes, including with, as you can see here, actually clear. And we will be talking a little bit more on that later on. We actually provide them singularly as well. If you already have a CI-600 or 602, we'd be able to get those for you. The 602 advancement is actually, it's it's 2 inches in diameter as opposed to the 2.5. Also watertight and insulated. The 2 inches actually fits into some existing installations for some older mini rhizotron systems. It's a little bit higher speed, so we're actually able to get scans at 100, 300, 600, and 1200 DPI, which has been actually a really interesting effect on some of the research done on mycorrhizae and pathogens. Now I'm just going to go over a little bit of how the systems work basically. You have the tube in the ground, the imager, you just dig a hole in the ground, install the tubes, and you want to allow the roots to grow around them. Once that has happened, you can insert the imager into the tube, begin scanning, and then the imager will actually go around at 360 degrees, do a full scan, and you can change the depths with it. Doing that, you're actually able to get a lot larger picture of the root system you're looking at. Now this is just going to keep replaying, so I'm going to go to the next. That kind of simple application allows a non-destructive testing for a lot of different applications, including root turnover, fine root growth, root hair formation, branching patterns and behavior, control versus treatment effects, root distribution, root interactions with like parasites and mycorrhizae and nematodes, and actually a whole lot more. So, we can do a lot of the application studies and tests here on our site, but a lot of this work has actually been done by researchers like you, and I'd like to kind of just show some of the examples. We do show some of these on our website. We try to get as many of them as possible, but so many are coming in where they're using, especially the 600, since it's been around for 10 years now. And now the CI-602, even though it's been around for only a year, there's already some requests coming in. But these applications are a wide variety. Here you can see they're across the globe from China to Spain to the United States. And they're interested in a whole host of different species and effects, including shrub species. You have a lot of grapes studies as well. Wetlands, this is able to be used in, and actually studies in permafrost and the tundra, which we'll see in a little bit. So being able just to use a fairly simple idea, being able to scan these roots, but at different depths, and doing it at a quicker, faster way and a better way of handling the data enables a lot more studies and a lot more work to be done in whatever field you're looking to do it in. Now I'm going to kind of cover the CI-600, 602 installation and some of the ways of methods of putting it, these tubes in the ground, mostly because that's most of the questions I end up getting right before going to RootSnap. So with the CI-600 and 602, we do suggest, if at all possible, you install root tubes during the previous season, just to mitigate as much of the detriment to the soil, the detriment to the roots that you're going to run to. Now, the plant by plant, it's actually going to be a bit different, of course, with your growing season. Some plants are very fast, rapid growing. Some plants just dealing with a poplar tree root issue. And those, for trees, are very rapid, but for most other plants, I wouldn't call that at all. But the more important thing is when you're installing these roots that you just remember that you want to keep the environment as similar as possible in the soil around the tube. So that means not allowing UV light in, not allowing moisture in that is atypical to what you would typically find in that area of soil, as well as actually any other like temperature and environmental effects. So when we do sell these tubes individually, and when we give you the 6 ones with the instrument, we actually have installation caps and give a bit of ability to buy a gas-powered auger to install these. Now, the other things I would definitely mention off the bat, just having run into this experience myself, is you want to make sure that the soil is not completely compact around the tube. You actually want to keep it about the same density as you would typically find around that plant. That way the roots grow as they typically would and not either bunch up because there's a lot of space and giving them a lot of space to grow, or completely avoid the tube because you've compacted the soil around and made it difficult for them to grow around the tube. And that being said, we have seen the 600s and 602s actually work in an entirely variety of different environments. This includes tundra, this includes wetlands, this includes very rocky soil, as well as very, very sandy or very clay-like soil as well. The setup can actually also be done to do repeated scans. Now, in this picture you see from the University of Copenhagen, you can tell that they're inserting the 600 into an already a tube that's been covered in, it looks like duct tape, just to make sure that UV light does not get in for their study. But what you can also do with that, if you wanted to do a singular location and just scan over a long amount of time, is you can actually just tell the software to do repeated scans, either 5 minutes, 15 minutes, up to actually 12 hours. So once you put that in, the actual scanner will just sit there, and every, say, 5 minutes, it would make a scan of the roots, and again for 12 hours. And you can do this as long as it's plugged into a computer and computers has power. So there are many methods I also want to mention of installation. We do typically suggest using a 45, 60, or 60 degree angle. This is a little bit for historical reasons, as well as the does keep out, especially in environmental, helps keep out things like rain and other environmental factors. But this instrument actually works either vertically or horizontally as well. So you can actually, and I'll show you in a little bit later in the presentation, you can really install it in a lot of very ingenious different methods. So, and just to give a good example, here we see tubes in a horizontal method. They have actually installed them right throughout the bed of these different boxes. And this is able to, you can take the scanner out, place it in one tube, do your scans, pull it out, place the cap back in, and take the scanner out as well, and just keep doing that repeatedly, fairly rapidly. Being able to get a full record of, especially in that box, of all of these root systems for all the plants right there. In the right, just kind of showing some of its abilities to work pretty much regardless of the environment. You can see it there being worked at on the tundra. The 600 is doing scans there, and we found that it's actually the acrylic tubes have more problems with the cold than the instrument itself and using studies. And then just bringing it back to kind of a success of different studies between wetlands and very dry temperatures. We have Dr. Murthy and Dr. Srinivas at the Central Research Institute for Dryland Agriculture in India. Here they're just actually studying root growth and development of 2 maize cultivars under 2 moisture regimes. You're looking at actually one moisture regime right now, and as you can see, they're covering part of the acrylic pipe with PVC pipe, just so, especially in those small biomes, you're not allowing the UV light in to keep the roots from growing around the tube. And here we see more of like an orchard application where we do have the 45 to 60 degree angles of tubes. But again, this is actually right around different pumps, right around in very moist, sometimes very dry environments from Demanio Zanatelli of the Free University of Bolzano, Italy. And it's just to kind of give some of the breadth of the applications. You can see that here he's studying this roots dormant during winter months, as you can see highlighted using the RootSnap software, and then activity and growth in the spring and summer. And being able to do that is just because you're able to take this instrument and scan in the same location repeatedly. And I'll show you a couple of techniques a little bit later on how to scan the same spots. But a lot of the work, so the scanner itself is really just for scanning and making sure you can do it out in the field. It is connected to a tablet that we do provide, and we'll show in a little bit. And the tablet actually will be coming with the software for both scanning, calibrations, as well as our RootSnap and image analysis software. This isn't to say that only the tablet can run it, it's just what we have found to be the handiest when you're out in the field. The image analysis software can actually put on pretty much any computer, as well as the scanning software. Now, the great thing with this is actually you can start playing with it today if you would like. We do provide it for free on our website, and we also include it with each one of the 600s and 602s. And we're constantly updating it. There's actually being some updates being pushed fairly soon within the next month by our software engineers. But it is a very easy and fast tool for analysis. Prevents backlogs of root images by being a lot quicker away than more traditional means. And it has a few features that I actually really enjoy. I think make root analysis a bit easier than I've been typically accustomed to. But then using the roots where you're mapping it out, it actually does a lot of automatic calculations. So you can get things like root count, total root length, total root volume, total root area, diameter, length, area, volume, and the average, window depth. And you also get the date and time of the images, so you can actually do a very quick study on what times, what variables that you might be running into. You do get actually a physical size of the image. I'm going to show you in a little bit, as well as being able to do scans and, as I said before, interlace them together to make a far larger picture. And then from that, you can actually get things like not just the individual roots, but overall systemic architecture for like branching angles, branch counts, and diameters of each one of the individuals. So let's take a look at what that looks like. Again, let's use Dr. Murthy's scan because I really like the coloring. And here you can see on the, put over here. On the left, these are actually multiple scans put together and stitched together using RootSnap. And so we will blow up one of these little sections just to show what they've done previously with their root scanning software and how it can look when you have mapped it entirely out. Now, as you can see, many individual roots are present in a single image. So for each root colored and mapped, the data that we've discussed in the previous slide has been recorded automatically in the software. And you can actually open that up and view it at your leisure. It does open up in like kind of a panel side. So you can actually just watch it change as you're doing your adjustments to your roots. So that is the bulk of what I was going to discuss with the CI-600, CI-602. We do have a whole range at CID Bioscience of different instruments to work with, so including the CI-110, which is a plant canopy imager, which we actually just gave a webinar about a month ago on. And the 202, the 203, both leaf area meters, the 340, a handheld photosynthesis system, as well as the assorted softwares actually for each one of the devices, and a CI-710, which is just a miniature handheld leaf spectrometer. So all of these are also available. I'm sure at the end of this presentation, we're actually going to be putting out a poll to see which one you would like another webinar on, and we'd be happy to have you vote on it. Now our Susie will be sharing a presentation on the video, just giving a kind of in-field representation of what it looks like if you're running this. and I'm going to give a little bit of a discussion and kind of explain what's going on. So again, we are seeing images of the CI-600. There you have to see like the locking nut to keep the serial port in place. Now, it's really being able to do non-destruction imaging in the field. Here, we're going to show you just all the pieces that come with the instrument, including the calibration tube. You have the scanner itself, as well as a slider rod. Actually, there's like a dozen names for this thing, but what it does is you can just attach it in to the scanner like so, slide it in, slide it out using the wheels attached to the scanner. And those rods actually have different links. So you can use these links with the cap that we also provide to kind of measure out as you're placing the scanner into the tube and then using it at different depths. And then it's pretty simple. This is actually the laptop right now that we are providing with the CI-600. And it's fairly plug and play. Once you do a calibration, this is the home position. You stick it into the calibration tube. And we do suggest that when you're doing your individual tubes, you actually utilize those two dots as well, kind of marking it at each one of the tubes. That way, when you're doing your scans and you're placing it in, you're just getting the exact same scan location every single time. It's not we find it just makes research and data a lot easier. It's not a complete detriment if you don't, because it is a 360-degree scan. These are some tubes we have actually installed in our own area. We do put kind of a foam core in to keep the UV light, but as you've seen, duct tape, PVC pipe, there's a lot of different methods to make sure that roots are growing in that area. And here, we're actually putting the locking cap on. This locking cap you use to both make sure that different environmental effects, UV, moisture aren't getting down into there and changing the environment, as well as you can actually lock it in place with the slider rod and just change the depths that you're using it at. And to scan, it's pretty quick. So here we have the RootSnap software in use. As you can see on the left, I have a certain set of image statistics set up, but you can use it just on your computer very easily using mouse. You can actually change the diameters just using the scroller if you'd like. The image as well, just so you can visualize better what's the roots. I prefer actually using the touchscreen very well because it's, especially when you're doing a lot of roots really fast, it is a lot faster method. It does do some thresholding, so you can kind of get an idea of how much roots are in the bulk of it. But also, as you're mapping it out, it is doing the selected points and taking all that data with it. So one of the things we wanted to thank you all for joining us today, because I know this is a bit of a bite of time out of your days, and we want to just make sure that we show our appreciation. We are offering, just by getting a quote today, actually 3 extra tubes for free to the additional 6. So that way you'll be getting 9 tubes if you ordered the CI-600 and through this quote. Now this is no obligation, it's just receiving a quote, getting an idea of pricing for your region. And just for that and for attending this webinar, you'll be able to get 3 tubes free. Now, to do that, Susie is actually going to be putting into the chat box the URL that you see ahead of you. So you'll be able to go through there and request a quote. I'm going to give you a few moments to do that, as well as go through some of the question and answers, and we'll start that next. And we have quite a few popping up. So I'm really glad to see this many questions coming out, and we'll try to get to as many of them as possible in the time we have. But I'll give you a few more seconds, actually another minute or so, to make a request if you would like, just clicking on the URL. And I am going to go over some of these questions real quick. I'm going to sign on in just a second. I just wanted to get a drink. Okay. Hope that was plenty of time. If not, absolutely no worries. You actually can use that link now that you have it pretty much at any time you'd like, and just sign on in. We'd be happy to help us. Oh, and we're going to go to the questions and answers. So the first question I have actually shows up. Are there any improvements on the speed by which digital images can be processed? This is actually one of the things that we hear a lot from students. So that even at the older technologies, the lowest DPI would take a long time to acquire data. The 600 has been around for 10 years. When originally established, you were using a lot older laptops, we're using older scanner technology. Since the subsequent update, since using laptops that are faster processing, we've actually seen the times decrease considerably, but really increasing the amount of scans you can do in one area. So I give an example. If you wanted to do like 100 DPI scans, honestly, in the work I've seen and the work of the researchers, you can get down to just a few seconds, to 10 to 15 seconds, just to do a full scan ahead. And then the higher the DPI, definitely the longer the scan is going to take, but especially with the 602, that has been, the time has been decreased. The next question, it'd be nice to know if any development done is the software to get the statistics of all the routes, including total length by single click as done in destructive route image analysis. So there has been some adjustments. I kind of went over a few of them to get the full information from the root system, but we are more than happy to add more. As I might have mentioned previously, our software engineers are currently developing another update for these root snap software, and any ideas, or we are definitely happy to entertain as that software is being developed. It's very important for us that it works as easily for you as we can hopefully get it for us, and once that's done, we will send that out to you for free. The next question I see is, does it produce 3D root system architecture? Right now, no, but that is actually a really good idea for future development. The next question I see is, how do we install it in, say, in a hydroponic setting? That's might be a bit more than I can go into detail here on this slide. The thing I do want to mention is that the instrument is actually very resilient to different environmental effects. With the caveat, do not just stick it straight into a water. Like most electronics, you don't want to just submerge the whole thing. But even if it's a high moisture, high humidity, we've had it at 95 plus and it works just fine. Just make sure that when you are installing it and you have those insulation caps, that you're making sure that the environment inside is the same as what you want that soil environment to be. It's going to be ubiquitous throughout. So that's maintaining temperature, humidity, UV, keeping UV light out. And we help that a lot with our insulation caps, as well as some other actual, in our manuals and online materials, some how-tos to fully do that. We have another question. Do you have to install before planting? I believe it's asking. And we do definitely suggest installing before planting. However, we have had researchers actually installing afterwards. It's really very much a kind of a plant-by-plant dependent situation. If you think your plant would be resilient to having an auger getting too close to it and the tube installed, and it would still grow around within a matter of weeks, that's going to be fine. If that's going to do detrimental harm to the plant structure, I would not suggest doing that. So ironically enough, in this case, what I usually find is like grasses are a lot hardier than things like trees or shrubs. Do you have microtubes to insert in pots 3 centimeters by 3.5 centimeters in height? No, we do not. Actually, the shortest tubes we have are 1 meter. We do actually provide, there are other lengths of tubes. I think you saw actually a few of them out there, but we definitely want to make sure that the full scan head, which is 34 centimeters by 6.35 centimeters, is able to be able to do a full rotation. Is there a special technique to install the tubes and insulate them to avoid moisture, dewdrops inside the tube, as well as supplement the images and give wrong readings? There are, again, these are very environmentally dependent, but the biggest thing we have found to mitigate that is just making sure that you have both the installation caps on if you're not near the tube, and if you're at the tube and doing your scans, to make sure you're having that locking cap in. That helps mitigate that quite a bit. Also, we found that there is different types of day depending on the environment. Say, for example, if you just have a rapid change in temperature in the soil itself, you're going to see a bit more moisture collection. Can this instrument be used for nematode analysis? I actually removed that from the presentation, very sillily of me, and the answer is yes. It actually has been used very frequently for nematode analysis, and especially since the 602 is now able to get down to a 1200 DPI, which is very high resolution, you're able to really start categorizing a lot of nematode cysts. Can you see mycorrhizae with the root images? Yes. I believe I had one picture on the slide that was showing some of the mycorrhizae as in root hairs, but yes, you can definitely get down to that level. Now, with those higher scans, I do want to say that it is going to take a little bit longer to do a full scan around, but you're able to see it. Is it practical in pots for alfalfa roots? Very much like the kind of the small beds I was showing earlier. We actually have had tested and had other researchers use it in pots. It just depends on how you put it in that acrylic tube. So if you have the tube go straight through the pot, have the pot here and have the alfalfa grow around it, that's one method. You can do it diagonally. Honestly, every time I think that we've run out of ways to do it, I see somebody else come up with a new method. The other next question, it looks like how many times can one tube be used? The tube actually can be used for years on length. The most important part is that that tube just doesn't get scratched up and damaged, so you're not really getting a clear viewing of the surrounding root system. Using acrylic, we have found that has mitigated a lot of the problems of past different substances. But if you have ground, like when you're doing your installation, that's when you're going to be getting most of the damage done. Once it's actually installed and you make sure that there's no water and the installation cap is on, you should be able to use that tube repeatedly pretty much as long as it's clear. If you do get water into it and say like you have standing amount of water, I definitely highly suggest don't put the 600 or 602 in there. That will likely damage it. It's kind of hit and miss. And what we do suggest is make sure that you drain out that tube if you're going to continue using it. So the next question we have is a 45 or 60 degree angle. What is the soil depth being monitored? So we do hand out 1 meter tubes. However, you can use even longer tubes than that. So you can actually just do a soil depth of, I would say 2 meters fairly comfortably, giving enough sliding rods. After that, you're starting to get really, gets kind of difficult to handle it just as a person. You need more like a machinations in place to do that. But you are being able to do scans at each one of those depths. So that way you can just, if you want to get a massive picture of what you're looking at, you can stitch all those scans together. So the image size taken in one scan, actually, that's just depending on the, well, that's actually a multi-part question, to be honest. It is measured by just the scanner head of the device itself. So as I already mentioned, it is actually just 34.3 centimeters by 6.35 centimeters of a scanner head on the 600. But when you stitch all those images together, you can get a far bigger picture, as well as when you're changing the DPI of it, that size of image is going to be the same, but of course you're changing the resolution and the pixel count. So it's, you can get a lot larger data files if you go to a higher resolution. There's another. How do you deal with air gaps around the tubes after installation? That's actually, depending on the soil, can be either almost a non-issue or an extremely difficult issue. And there's actually a lot of methods online. I would definitely suggest starting either with our website and seeing some of the ways that it's been applied by other researchers, or even check out, so like the University of Copenhagen or the Permafrost Institute. They've had a lot of issues in dealing with that. Or I'd be happy to take us online. We are offering consultations on the side and be happy to discuss that. All right, the next question is, did you experiment with this technology with vegetable crops? So yes, actually, as you saw in India, it's being used with maize. In the picture, I believe it's still on the screen, it's being used with corn and corn rows, but it's also been used on squash, beans, tomatoes. Actually, the list is very, very, very long. Since the 600 has been in use for over 10 years, it's been well documented. 602, it's only been around for about less than a year now. But it is a fairly similar instrument, and it's getting very similar research. The next question is actually a very good one. How often do you have to calibrate? So you don't have to calibrate after each measurement. When we do request, you just stick it in the tube, press calibrate on the screen, and do that either once a day if you're staying at the same DPI and fairly similar temperatures. If you are changing temperatures, like going, say, from one acrylic tube that is at, say, I'm from America, so it's going to be 80 degrees Fahrenheit, and then going to another one where it's, say, 50 degrees Fahrenheit of the soil, you're going to see that the acrylic tube itself is actually going to change its diameters just slightly. So we actually provide a way to adjust the scanner head on the device itself. It's really simple, just adjust the screws. The scanner head will raise a little bit or lower a little bit depending on its connection with the acrylic tube. We make that really easy, but after that, you definitely want to calibrate again. So I would say keep that white calibration tube with you. And to calibrate, it's really easy. You just stick it in, make sure it's in the home position, and press go. Also, if you're going to change the resolution, so say if you want to do the initial soil scan, the topsoil, you're wanting to do 100 DPI because you just want to get an idea of where the roots are there. But the lower you go, you know that's where you're going to want to see like your mycorrhizae, your fine root hairs, your parasites. So at that level, you want to get more to like the 600 and the 1200. To that, you would have to scan at the 100, pull it out, do the calibration at 1200, and then put it back in. Thankfully, because you can measure it out, you'll be able to know where those screens overlap, and you'll be able to tie them all in very quickly using RootSnap. The next question is, will the tube influence root growth? So acrylic tubes have been long studied, and installation is what, and how it's installed is the biggest factor for affecting root growth. Making sure that you have the environment and the soil around it very similar to the roots that how it was growing on the rest of the plant is very important. And that means make sure the soil is not compacted, making sure that the soil has about a similar moisture rate. You're not having larger amounts of holes in it, and you're also being able to cut out and make sure UV light is not getting in the tube. So the insulation caps help with the UV light, though I definitely suggest if you're having large parts of the tube stick out of the ground, make sure that those are wrapped up either by, some of the examples have been PVC, duct tape, really, or even you can do white paint, but just some way to keep out the UV light from getting down into the ground, inhibiting the root growth. The next question is, how much faster is the 602 compared to the 600 at the same DPI? And in trials, it's actually been about twice as fast. We found it's a little slower when it's cold, but it's about, it moves in a lot faster pace than the older 600s. Actually, more updated 600s are moving faster, but as technology increases in one, technology increases the other. Can you use RootSnap for root images obtained by other scanning devices? The answer is yes. Actually, you can import really any image into RootSnap. It's not going, if you import a image that's not roots, of course, it might have some issues simply because it's trying to delineate between root and soil a lot by color contrast, light contrast, and brightness. So it's not going to be, if you're trying to put your picture in it, it's going to come out a little funky, but you'll be able to use it for other pictures. What is the recommended DPI for studying root hair and mycorrhizae? Ooh, 600, 1200. I've seen because the CI-600 does go down to 600 or 900. I would say keep around there if for larger colonies, but yeah, I would definitely look into using the 1200 DPI just so you can get the full, as good a resolution as possible in your studies. Let's see, how does it work for bigger roots, perennial trees? Because of the scanner head, actually it works fairly well. Of course, when you get it really depends on the root size. If you're thinking about roots larger than, say, a couple inches, you're only going to be getting them at a glancing angle to the tube. So you'll still be able to see a few, but you're not going to see nearly as many as you would of like the smaller root structures and systems. Currently, next question is, what is the technology used in the scan? If it's the visible light? Currently, yes, though we are actually looking into further technologies, including different LED pigmentations, some light NIR, not too far into the range, and also some UV, and seeing how that can be readily added to the scanner. We're not ready yet, but we are researching it. The next question is, can I have a copy of your PowerPoint slides? And the answer is yes. Actually, once we are finished with this presentation, we're going to take a recording, make it available to everybody, as well as take the PowerPoint and be able to get it out to anybody who would like to take a look at it. The next question is estimating nodule size and number. You can kind of do that with RootSnap, that is correct. And you can, just by adjusting, say like which color, which one are those lines and the diameters, you can get a good size, diameter, overall mass of your different nodules. So the next question is the technology able to detect and identify root disease? Now the answer has been yes, it has been able to detect it. It is just a visual scan, so it does require an expert right now to be able to identify, is that a root disease? Is that discoloration, or just discoloration from the scan or the surrounding environment? It still requires, the human touch, but we are able to, pick up root diseases of a very large amount, just simply by being able to scan in areas where you typically couldn't see. So the next question is, how long can a tube last in the outside environment, especially under freezing conditions? That's a good question for acrylics. I know the CI-600, the scanner itself, has been used throughout an entire day long of permafrost studies. So I know that will work. With the acrylic tube, I would have to ask some of the experts in the field regarding how long they stand, but I do think it's from years. My interest would be decades, but we definitely know it's over. If you're looking at 2 different seasons, you'll be absolutely fine. The next question, is there a special application to suggest the CI-600 to a customer? We do have actually a couple different reasons I would suggest the 602. One is that the 602 does go up to 1200 DPI and is a little bit faster scan, so you can actually be able to do more rapid scanning with that and at higher resolution. So if you really need to do the parasite studies, mycorrhizal studies, a lot of fine root hairs, you can get it with the 600 at the very edge, but you need to, with the 602, you can definitely you have multiple choices of that resolution level. The other thing I would suggest is because it is a thinner, it does fit, there's a lot of 2-inch diameter tubes out there already from older technologies, and it does fit in those if you have them already established. Let's see. The next question is there a miniature version available? And currently, sad to say there is not, but that's something we can definitely keep in mind as we continuously progress the advancements of this technology. And the next one is how sensitive the cameras are to soil types. Actually, so the camera itself, as long as you're making sure that scanner head is up against that acrylic tube and the acrylic tube's not deformed or scratched up itself, very, very sensitive and able to pick up your different soil types. You can see each one of the visible scans. So you can see very rocky, very sandy, very clay, as I typically find where I'm usually from. But you can actually definitely pick that up pretty readily with the scanner. Now, using these tubes, since they are acrylic tubes, they are fairly resilient, but the next question is dealing with them deforming below ground. And the answer is anything. There's a very little bit in the ways of a clear tube that can't deform below ground at this point if you're giving it the right conditions. So if you're flooding the ground, if there's a lot of pressure. But the biggest thing I find is the installation, the initial installation, that really dictates how well that tube is going to perform as you're using the scanner on it. Usually after being installed and after it's settled and the roots have grown around it, those tubes are pretty much good to go for the long haul, for actually many years of study. The next question is, can we still have the option of Win Rhizotron software besides the root snap? And the answer is yes. Actually, these scans are all being saved in a couple different methods, both with the data alongside as well as just a simple image. You can take the simple image and put it in Win Rhizotron or any other software you'd like. The next question is, what does the tip of the tube look like? Going to say, gesture towards the screen, you can see we actually have insulation caps on both ends, and that's what it currently, both of them, you see the large diameter for the 600 and the smaller for the 602. The next question, is it possible to measure cassava roots, especially necrotic tissues and cassava roots? So personally, I know there's a research already been done using 600 on cassava roots. However, I don't know if it's been done for necrotic tissues. I would imagine since it has been done for necrotic tissues of other plant species, unless there is some kind of variable I'm not aware of, it should be able to be useful for that use. All right, the next question is, any reason for not being able to save a traced root image for later viewer analysis on the computer outside the software? Actually, that's a good question. I think one of the more recent updates with the software has been to be able to save those traced images as actual just images. So you see the tracing simply because I was just able to do that for this slide presentation. But if you're asking for like the data associated with it, that would be part of the software since it is a lot more data and needs to be handled differently. I'm going to answer a couple more questions. However, we are getting to about 8:55 my time, wherever that is globally. So we're getting to run into the other part of the hour, and we still have a lot of questions coming in. So what I would like to do is just make sure is like emphasize that I am available and my compatriots colleagues are available for a discussion on your individual projects, and be happy to consult with you. There is a link that I think Susie should be adding into the chat fairly shortly that you should definitely check out, click on, and set some time aside so you can talk to myself or Katrina, and we will be able to help you with any of your projects, especially when using with the 600, 602. But I will answer a couple more, and then we'll probably close this out. And yeah. Okay, we have a lot more questions coming in, actually. So once I answer a couple more, we'll close this out and then end the presentation, and we'll share this all with you by later today. All right, the next one is, how long does it take to get the total route length of a typical single depth 360-degree scan? Actually, it's very, if you're talking about the full scan, you can get it really depends on how complicated the root structure is, to be honest. If you have a fairly simple root structure, I've seen it done within 20 minutes or less. If you have extremely dense, very complicated, and you're mapping out each single one, that is always going to be a longer affair. So but it does do some automation in the software itself. So when you do, say, a few lines, you're gonna do automate, and it will try to estimate by the pixels how long those roots really grow. I find that very useful. Towards the ends, it gets a little wonky, but you just snap them back into place real fast, and that makes the process a lot faster. Again, next question is, do we need to calibrate on various DPI? And the answer is yes. You want to calibrate before every single, if you're changing the DPI, you do want to calibrate again. But it's just as simple as pulling it back out, knowing where you had the depth before just by using the links of the rod, putting it into the calibration tube, scanning, calibrating, and then pulling it out, putting it in for the resolution you're looking for. And the next question is, can software automatically log and save the root readings? And the answer is yes. It does log them, does save them, keeps them in a root scanner file, which you can open up later on. Root snap file that you can open up later on at any time. You also can save the images themselves, so you can open up another software or just view as you would please. The next question is, can the image analysis software recognize roots? Traditionally, taken a long time to manually select roots and measure the lengths in images taken at certain time intervals to get root growth. Again, the image analysis, we have developed the automatic selection a little bit, so you're actually able to start, especially the selection, and then just click automate, and it will continue on through the similar size pixels. Once the pixels get too different in their coloration, brightness, intensity, it will stop. But then you can, and if that is not exactly on your root, you can really quickly just point, drag it over, and it'll snap back into place. So the next question is, again, how big of the area can be measured? That is simply the scanner head and dimensions. So you have 34.3 centimeters of the scanner head. You can scan that and just keep doing that for like 6 degrees of diameter, 6 centimeters of diameter along all the way through. And you can do, I would say, 2 meters very comfortably. And I've known some researchers to do more. And then the other question, actually, and just I would say we will finish this off with, is we do have another webinar being presented today for this. It's the same one, but if you were unable to attend this one in time, you can actually sign on to that one. It's at 6 p.m. Pacific Standard Time, and we will be able to give the same presentation as well as answer all the questions that you have available there. But again, my name is Dennis Fisher, and we have, if you have any questions following this, please join on to the consulting link that Susie has provided, and you'll be able to set up some time with me at your leisure, and we'll be able to discuss your project or how you would like to use the CI-600 or 602. We can also discuss a lot of other applications and instruments because the CID line is fairly broad. Now, I believe Susie is going to put a poll for what instruments you would like to see a webinar on next, and then we will wrap up this webinar. So go ahead and vote what you would like to see more of a presentation on, learn more about, and that way we can focus our efforts in the next one coming up probably within a month or less, and be able to get that information out. Sorry, actually, and also with the voting, you just want to make sure that the URL for requesting quote is also in the chat as well, in case you would like to do that. And again, definitely want to say that if you use that particular link, you'll be able to get the 3 free tubes for free whenever you or your university or group are able to make an order for the CI-600. This is just for us to make sure that you are able to attend to the webinar. And we want to make sure that we're showing that we're happy you were here and we can give you 3 free tubes. And again, I would like to thank everybody for coming and look forward to talking to you in future webinars.